Okay, so we're going to talk about enzymes. And we're going to talk specifically about types of enzyme catalysis. Okay, so there are 5 major classes of enzyme catalysis. So the 5 classes of enzyme catalysis that we'll talk about, one is called general acid, general base catalysis. The next is called covalent catalysis. Where there might be some sort of covalent intermediate during the catalyzation of a reaction. The next is called electrostatic stabilization. Another is called proximity effects. And then the 5th is called preferential stabilization of the transition state. And then this T with the cross across it stands for the transition state. The transition state is also at the top of this hill of the activation energy. It's the hill that you have to overcome to then go down in free energy. So this is called the transition state. So stabilizing the transition state kind of gets you to the downward part of this reaction quicker. Okay, so we are going to talk about these types of catalysis. And most enzymes will use multiple of these. It's not that an enzyme only uses one or another form of catalysis. There are a lot of examples in the lecture slides, and I'm going to go through one specific example, and that is with the enzyme chymotrypsin. So chymotrypsin belongs to a class of enzymes called serine proteases. Serine proteases. So serine proteases are a type of enzyme. It's a protease. Prot means protein. Serine because there's a serine in the active site. So this is a serine protease. It is going to cleave the peptide bond of proteins. There are many different types of serine proteases. There's actually a list of serine proteases on slide 43 of your PowerPoint lecture. There's, 10 or 12 serine proteases there. They're found in all different types of locations: pancreas, blood, serum, cells. We are going to talk about chymotrypsin. Chymotrypsin is actually made in the pancreas, and the purpose of chymotrypsin, which is what we're going to specifically focus on, chymotrypsin it's the digestion of proteins. So digestion, it just literally means it's going to break apart the proteins, and it's going to do that by cleaving that peptide bond. So chymotrypsin All serine proteases have what's called a catalytic triad. So all serine proteases have a catalytic triad. Triad just means 3. So that means there's 3 residues in this protein. Remember, chymotrypsin is an enzyme. Enzymes are proteins. So there's 3 residues in the active site of this chymotrypsin that are going to be responsible for the cleaving of the peptide bond. So one thing that is important is there's that big list of serine proteases. And the reason that there were so many serine proteases is because these serine proteases are specific to different types of proteins. And they're going to cleave the peptide bond at specific locations. So chymotrypsin, for example, cleaves the peptide bond on the carboxyl end of bulky hydrophobic R groups. So some of these big bulky R groups that would fall into this category, these are a few: methionine, phenylalanine, tryptophan, tyrosine. These are where the chymotrypsin would cleave. It would be on the carboxyl end, on the C terminus, after these big R groups. There are some slides that go through what I'm about to explain in my drawings, and the slides do a much better job illustrating it. This is slides 45 and 46 in your PowerPoint lecture that shows the active sites of all of this. So what's important for this specificity, I'm going to draw this. So I'm just going to draw the active site. So we'll say here is just the active site. So all of this would be chymotrypsin. And this is the active site. These right here, these 3 locations, this is where the catalytic triad is. Right above the catalytic triad is called the S1 pocket. And this S1 pocket is specific to whatever group this enzyme is specific to. So for example, chymotrypsin cleaves after big bulky R groups. So that means chymotrypsin has a great big S1 pocket for these big bulky R groups to fit into. If you look back at the slide that talks about all the different serine proteases, you'll see there's all kinds of other ones. So trypsin, for example, not chymotrypsin, this is just another example. Trypsin cleaves after positively charged R groups like lysine and arginine. So trypsin's S1 pocket is going to have a negatively charged amino acid in here to stabilize that positive amino acid so it can cleave here. This is where the cleavage is going to happen at the catalytic triad. The S1's pocket is what gives the specificity to the serine protease. Elastase is another serine protease, and it cleaves after small hydrophobic R groups. So like glycine, valine, alanine, leucine, isoleucine. So that means its S1 pocket is going to have hydrophobic amino acids in it. So the S1 pocket is what gives the enzyme its specificity. There's no chemical reaction happening here. There's no cleavage happening there. The cleavage of this peptide bond is happening here at the catalytic triad. So again, this is drawn much more neatly in your PowerPoint slides. I'm just going to use this to kind of talk it through. So here, we'll say this is the serine, and then here is a histidine, and here is an aspartic acid. So this is the catalytic triad: serine, histidine, and aspartic acid. And these three are in all serine proteases. So every serine protease has this catalytic triad. They all have different S1 pockets right next to the catalytic triad. And that allows for the specificity of the exact location of where that serine protease is going to cleave. Okay, so what happens, I'm going to draw very roughly the R groups of these amino acids. And again, this is much better on the slides. So here is serine. Here is histidine. And then down here is the aspartic acid. Okay, so what's happening here is, again, look at the slides. Look at slide number 45 for this. But what's happening here is the alcohol on the serine is going to be transferred to the hydrogen on the protein that's coming in. So you have a protein that's coming in, and the protein is going to have, again, a big bulky R group in here. And this is where the cleave is going to happen. So the alcohol on the serine right here is going to transfer a hydrogen, and it's going to form a covalent intermediate with this peptide here. And when it does that, it's going to make this planar, and it's going to attack the carbon in this peptide, this chain. Remember, this is a polypeptide chain. It's going to attack that carbon because it's created this temporary, what's called an alkoxide, and that's a strong nucleophile. So it attacks the carbon of the carbonyl, and it produces an unstable tetrahedral intermediate, and now this polypeptide is no longer planar. This is going to cause the polypeptide chain to be cleaved. It's going to be cleaved here. So that quickly collapses, and it breaks the peptide bond. So one thing that I maybe haven't said directly, but that is true of all catalysts, not just enzymes, is that catalysts are not consumed in a chemical reaction. So while the point of this reaction is to cleave this peptide bond, it also has to regenerate this active site. So that also is going to happen. So I'm just going to walk through all of the steps of what's happening here. So the polypeptide substrate binds non-covalently. It bonds non-covalently in this hydrophobic pocket. So your textbook calls the S1 pocket the hydrophobic pocket. It happens to be hydrophobic if this amino acid is hydrophobic. It's not here because I drew a different one. But this S1 pocket is specific to whichever location that it's cleaving. So the hydrogen is transferred from the serine to the histidine here, and it forms an oxyanion intermediate with the enzyme. The peptide does. Then we have what's called an acyl enzyme intermediate. So the hydrogen transferred to the carbon terminal fragment is then released by the cleavage of the carbon-nitrogen bond. So remember, the carbon-nitrogen bond that's cleaved, this is a peptide bond. The peptide bond is cleaved, and the N-terminal peptide is free. It's removed. After that, a water molecule comes in. Remember, this is all happening in an aqueous solution. The water molecule comes in and it binds to the enzyme in the place of the departed polypeptide. And then the water molecule transfers its proton back to the histidine that was removed. And then the hydroxyl group is going to go back here to the other substrate fragment, and then the tetrahedral oxyanion intermediate is formed. And then the second polypeptide will leave. So this will leave as one fragment, and then this will leave as another fragment because these two pieces were cut apart. So this serine protease uses a general acid, general base catalysis by the transfer of this hydrogen from this histidine, acts like a general acid by donating a hydrogen. It also uses covalent catalysis because there's a temporary covalent intermediate formed between the serine and the peptide chain. And it also uses electrostatic stabilization in the charge interactions that's going to hold this polypeptide in place. So chymotrypsin uses 3 of these 5 types of catalysis in this mechanism of cleaving a peptide bond. There's a lot of examples of this, and I included graphics from multiple textbooks showing these processes, and some of them have the active site kind of flipped around. It's the same process, it just was done in a different order in a different textbook, so don't be confused. Just look for the serine, histidine, and aspartic acid. In the second set of slides that I've posted about them, they're just flipped over. So what's important for you to know about chymotrypsin is that the specificity of the location of the cleavage of the peptide bond comes from this S1 pocket. And for chymotrypsin, this would be a big pocket because there's big bulky R groups that are going to fit in there. And then the coordination of the catalytic triad, the serine, histidine, and aspartic acid, and how they are all coordinating this to form an attack on this peptide bond, cleaving this, and then eventually a water molecule will come in after to restore the histidine and then also donate a hydroxyl group to the other end of the polypeptide.